students, I'm Madam Joyce from Educator Z. I'm teaching biology, and today I'll be with you, students, to discuss our topic, which is coordination and irritability, which one is the most easiest topic in the Form 5. And here we are going to discuss on our subtopic, which is the sensory receptor. So my student, before we proceeding further, we have to recall ourselves on where we end on the last session, is whereby we try to look on the structural difference between the rods and the cones. Thereby I provide for you a summary of the differences between the between the rods and the cones. We have seen almost 10 differences between the rods and the cones in terms of the structural difference. So my student, in our today's session also will proceed looking on the mammalian eye, but here specifically we are going to look on the mechanism of photoreception. We are going to see mechanism of photoreception. So before we are going to see what is photoreception, before we are going to see the mechanism, we have to know first what is this photoreception, and thereby we're going more further to discuss the mechanism. And here we are saying that photoreception is the process by which light is detected and converted into a signal that can be processed by the nervous system. Meaning that hizo signals na be converted kutoka kwenye ile light signal into light signal, or we can say that electrical signal, that it can be easily interpreted within the brain. Is due to the presence of this photoreception, the process by which the light is detected and converted into the signal that can be proceeded in the nervous system. So how does this signal converted from the light into a signal that can be converted or enter through the nervous system? Is when now we are going to see the mechanism. Remember, we are discussing on the mammalian eye. So the mechanism of the photoreception, now we are saying that rods contain a light-sensitive pigment that is called the rhodopsin. That is the rhodopsin, which is attached to the outer surface of the vesicles. Kwa hiyo sasa within the retina is where now the image can be interpreted. So now we are saying that within the rods is where now we have the pigment which is very sensitive to a light, which is called the rhodopsin. This rhodopsin, it is attached or it is embedded in the structure or in the outer surface of the vesicles within the rods. So this rhodopsin is a molecule formed by a combination of a protein called the scotopsin, with a small light absorbing molecule called the retinene. So tunasema kwamba hii tunayoita rhodopsin. So the rhodopsin here, we can say that it is a molecule formed by a combination of two proteins. Protein ya kwanza inaitwa scotopsin na ya pili inaitwa retinene. Ndio ambayo tunaenda kuform hii structure au hii pigment ambayo inapatikana ndani ya rods inayoitwa rhodopsin. So now what is this retinene? Retinene is a carotenoid molecule derived from vitamin A. So hii retinene ni molecule ambayo imetokea kwenye vitamin A. So vitamin A ndio ambayo ina bring about retinene. So now when the rhodopsin molecule is exposed to a bright light, it breaks down into a retinene and scotopsin. This process known as breaching. So hapa tumepata structure. Tumesema kwamba rhodopsin inatengenezwa na two molecule of protein, that is scotopsin together with the retinene. So when that rhodopsin it is exposed to a light, ipo kwenye mwanga tu kidogo, ndipo ambapo tumesema rhodopsin inakuwa ni high sensitive to light. Ndipo ambapo inaenda kuform ina break down to form the its constituent, that is the retinene and the retinene and the scotopsin. Hiyo process naita breaching. So what is the breaching now? So the bleaching here, we can say that is the process of breaking down the rhodopsin molecule into retinene and the scotopsin. So the bleaching is the process of breaking down rhodopsin into scotopsin and the retinene. But this process it is aided by a breaching process. As you can see here, we have this rhodopsin, but the process of breaking down this rhodopsin to form retinene and scotopsin, it is called the breaching. So why is the rhodopsin it is breaking down? Is due to exposure to light. Pale ambapo itakuwa patatokea na mwanga kidogo, tumesema hii rhodopsin pigment it is more sensitive to a low light content or constraint. So hapa tunasema the presence of light will breaking down this molecule of protein that is called rhodopsin into retinene and scotopsin. So now we can also say that the scotopsin, or we can say that rhodopsin is reformed immediately when the light stimulation decreases. Kwa sasa inakuwa ina reform again pale ambapo mwanga utakuwa unapungua, meaning that ina reform again, inarudi kwenye its original shape. So since after breaking down of the rhodopsin, we get retinene and the scotopsin. So after the low light content, the retinene and scotopsin are united to form the structure inayoitwa rhodopsin until there is a presence of low light content. So now we can say that retinene is first converted into a cis retinene and then it combined it to scotopsin. So this process is called the dark adaptation. So remember, all of these breaching processes and so on, they are taking place during a dark time, since this rhodopsin it is found within the rods, and the rods they are very sensitive during small light content, or during dark content, or during night. So that's why we can say that that process it is called the dark adaptation. That is, tumesema hapa ile retinene first ina be converted to form a cis retinene, then ina recombine with the scotopsin, ndipo ambapo inaenda kutengeneza structure ambayo inaitwa retinene, inaitwa rhodopsin. Na hiyo process nzima ya kureform back the rhodopsin ndio ambayo tunaita dark adaptation. So make sure that you underline this word, dark adaptation. So that process which are occurring within the rods. Similarly, we can say that the cone system has very high spatial resolution, but it is relatively insensitive to light. Hii ambayo tunaita cones, it is less sensitive to light. Yani yenyewe haijawa bado sensitive to light yoyote, because these cones they only function during a light or bright day or bright time. So now we can say that it therefore now specialized for acuity at the expense of sensitivity. So the property of the cone system allows us to see the color. So hile tunayoita cones inakuwa na high visual acuity, the ability to see or to distinguish different or fine material. Vitu vidogo vidogo sana, we can have that ability to distinguish is due to the presence of cones. But not only that, also these cones allows us to see color. Nowadays we can see different kinds of colors. We can see this is black color, this is white, this is green, this is red, this is blue. It's due to the presence of cones which are found in our eyes. So my student, thereby you have to know that in explaining the physiology of in explaining the mechanism of perception, photoreception, you have to explain both cones and the rods. whereby within the rods you have to explain the breaching process, how it occurs, and the reformation of the rhodopsin again. So also you can ask yourself that why the retinene change their structure, that is from cis to trans, is due to the reformed back of the structure, which is called the rhodopsin, that is found within the rods. But within the rods, within the cones, tumesema kwamba yenyewe inakuwa ni insensitive to light. Kwa hiyo sasa inakuwa na high visual acuity, but also these cones they have make us have ability to see different kind of color within our environment. So my student, now let's proceed in the next part of this session. Is where now we are going to see the physiology of seeing. Remember, we are discussing on the mammalian eye. So thank you for listening.